In this video, we will discuss about the mechanisms of how these TGF-β induced cells prevent inactivations of Rac1. And from the heading, it's clear that TGF-β downregulates ARHGAPs to prevent inactivations of Rac1. How they come up to this point, we will discuss that thing in this video and conclude this paper. So, in the previous videos or the results that we have understand in the previous video shows that this TGF-β downregulates cells will prevent this inactivations of Rac1, and that thereby it will cause cells to have more motility. But in the case of these SMAD3 knockdown cells which are infected with this mutated SMAD3, we see more activations of this Rac1. So what they did is they did the transcriptome analysis of the TGF-β induced cells, either of the SMAD3 knockdown cell lines or this A59 SMAD3 knockdown cell lines that are infected with this mutated SMAD3 genes. And what they are able to find out is that there is the downregulations of genes that are observed in the SMAD3 knockdown cell lines, but those downregulations are not observed in the SMAD3 knockdown cell lines that are infected with this mutated SMAD3. So from that they finally come to a point that they need to check this kind of proteins, that is ARHGAP 22, 24, 27. They check these three proteins, and what they are able to find out is that in the case where these proteins are downregulated, what they are able to see is that in the wild type case, they are able to see that there is a so they check this ARHGAPs upregulations or downregulations in the TGF-β induced cells at different conditions. So in the wild type TGF-β induced cells, they are able to see downregulations of this ARHGAP 22, and for the case of this SMAD3 knockdown, they do not see any downregulations. And when this A549 SMAD3 knockdowns is infected with this SMAD3, they again see the downregulations, and when they are infected with this mutated SMAD3 knockdown, they do not see downregulations. So one thing, let me just correct myself. So transcriptome analysis are done on TGF-β on this SMAD3 knockdown cells that are infected with this SMAD3 or with this mutated SMAD3, and they are able to see that these ARHGPs proteins are downregulated where the wild type SMAD3 is infected, but not in the case where the mutated SMAD3 is infected. So from that they wanted to find out the impact on these ARHGAPs on the TGF-β induced cells. So we can see that the downregulation is clear when the wild type SMAD3 is infected, but not in the case where RKPs. This indicates that TGF-β helps in the downregulations of these ARHGAPs proteins, and that plays a role towards these ROCK inhibitors that inactivates or that regulates the ROCK inhibitors. And for this ARHGAP 24, they see the similar trend, and similar trends are again shown for this ARHGAP 27. And along with that, they also see for the control, they see this SER PINE1s, and they see that these control signals are in line with all the other conditions, that is where the this mRNA is not dependent on this mutated SMAD3. And once they find out that the TGF-β induced cells downregulates these ARHGAPs, they wanted to see how these downregulations can be inhibited. In order to inhibit these downregulations, they use cyclohexadimine treatment that prevent the protein synthesis, and with this they are able to regain this they are able to prevent the inhibitions of these ARHGAPs. that can be seen in the panel B, where we can see that cyclohexadiamine treatment will cause more ARGAPs formations compared to the case where no cyclohexadiamines are present for all the three cases ARGAPs 22, 24, 27. And once they find out that how they can prevent the inhibitions of ARGAPs, now what they did is they wanted to check the impact on the cell motility. They wanted to check the impact on the cell motility by inhibiting these ARGAPs in the TGF-β SMAD3 knockdown cells that are infected with the mutated SMAD3. So first, the panel C, they shows the effect of the silencing mRNA against ARGAPs 24, 25, 22, 24, 27, and they did the experiment on the 24, where they can see that there is down regulations of the ARGAPs mRNA of this 24 number, and they can see that when this silencing mRNA is used, they see a decrease in the when the silencing RNA is used, they can see a decrease in the mRNA of the ARHGAPs. So now they check the impact on the cell motility, and they are able to see that the cell motility seems to be recovered, where ARGAPs is been down regulated in this A549 SMAD3 knockdown cells that are infected with this mutated SMAD3. We can see the cell motility is being recovered, whereas in the control case where the ARGAPs 24 is not down regulated, cell motility is not recovered. Now these things are quantified in the right hand side of the panel D, where we can see that when this ARGAPs 24 is mRNA is used, that is to down regulate this ARHGAPs in the A549 SMAD3 knockdown cells that are infected with the mutated SMAD3, they are able to see around 60% recovery of the migrations. This indicates that ARGAPs 24 plays an important role towards preventing the cell motility, and it is and there are other ARGAPs that could also contribute towards the same phenomena. So this is what they finally conclude there. I hope you understood their approach to confirm this hypothesis. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.